Hi there and welcome to lecture 4. We're now going to start to move more into enzymes. So we're moving away from proteins as a group. We're going to go more specifically to talk about enzymes. We're going to talk about how enzymes enable all of the biochemical reactions. So we're moving our way now towards metabolism. In terms of the study aims for the lecture, you need to understand the properties and the structure of an enzyme, enzymes, describe how enzymes actually facilitate reactions. We'll talk about different models, so the induced fit model that allows substrate binding to what is called the active side of an enzyme, and we'll also compare and contrast different types of inhibitors. And the two main types are competitive and non-competitive. These in particular are going to be important when we come to practicals, particularly the last prac 6, where we look at enzyme kinetics, because you'll be using inhibitors. So it's really important that you understand how an inhibitor can work and how we can identify these changes in enzyme parameters. So space for your questions, and obviously the lecture in the notes. Okay, so we know that within a cell, within an organ, within the host, that the body is trying to maintain homeostasis, is trying to maintain a basal level. But we also need to respond to stimuli, and that just might be something as simple as light, okay, or hunger, or a pain sensor. So as a result, to do this, biochemical products must be produced fast enough, so at a rate that meets the need of the cell. If it's too slow, that could lead to cell death, because the cell is not able to respond effectively, and that's going to lead to the death of the cell or even just the death of the organism. So it needs to be fast enough. So as a result, these products have to be created through a series typically of chemical reaction pathways. So what this means is multiple steps. And we'll start off with single steps, but multiple steps are really just linking together lots of single steps. And typically for this to occur, we need to use a catalyst. Now, from a chemistry perspective, a catalyst is a chemical or a reagent that accelerates a reaction but is not consumed in the reaction. Okay? So it facilitates it or speeds it up, but doesn't get used up. Cells use enzymes as biological catalysts. So they will accelerate the biochemical reaction. They're not consumed, they allow it to occur. Why do we need a catalyst? Because without these enzymes, these biological catalysts, some reactions might occur too slowly, as we set up here, or they may not occur at all. So without that ability, life would not be possible. So very, very important groups of molecules in class. So what are some of the general properties of enzymes possible? So very, very important groups of molecules in class. So what are some of the general properties of enzymes? So as I mentioned, as catalysts, enzymes are not consumed by a chemical reaction. That's fine, we've covered that up there. Most enzymes are going to be proteins, most, but there are exceptions. So within the ribosome, you have a molecule called a ribozyme, and this is actually built from ribonucleic acid that has enzymatic activity. But I think apart from this specific situation, you could probably assume that most of the time we're talking about proteins as being the enzyme. Okay, it's going to be more of the exception when we talk about things that are not proteins with enzymatic activity. Enzymes act on substrates. So this is what they are targeting, the starting molecules, and after that they lead to the formation of a product. So it is the end result. These are two sets of terminologies, so substrate and product, you must become very familiar and comfortable with, because we will talk about, the substrate for this reaction is this, and the end product is this. And then we'll talk about how the product can then actually go back and affect the enzyme. So it's important that you understand which is which. Enzymes are very specific to a reaction, and so they will catalyze just that individual reaction. So what that means is that side reactions are very rare. If we contrast this with, say, normal laboratory chemistry, this is not the case. And you would have learned this in first year, where you say we've got A plus B gives us C, but in small amounts we might also get D and E. And we don't want these, but it's just part of the reaction. This doesn't occur in an enzyme. An enzyme will just give you this. You will not see these side reactions, so very, very specific. And of course, this makes a lot of sense from a biological perspective. And lastly, enzymes function within a defined temperature and pH conditions. So they have what are called optimum conditions. So if we look at enzyme activity on the vertical axis and temperature, you can see that as temperature goes up, activity goes up, and then it goes down. What's happening here? This is denaturation. So if this is, say, 37 degrees, this might be 40 degrees, 45 degrees. So we get too far and it denatures. You can see that it doesn't take a lot for all of the protein to be denatured. So you want to always be somewhere at the optimum concentration, but at the lower end as opposed to the upper end. And if you look here are two examples of two enzymes, pepsin and chymotrypsin. These are digestive enzymes. So you see these in the stomach. You can see that in terms of activity, pepsin, its maximum, it's not very straight, is about 1.5. Chymotrypsin is about 8. So quite different. So these will still work at, say, pH 3 or pH 3.5, but notice it's about a third as active. So these are the optimum temperature and pH for that particular enzyme or enzymes. Enzymes also catalyze a whole raft of chemical reactions, and we can break these down into 6 classes. You will see these time and time again. So I want to just spend a little bit of time talking about these, but I also want to take this opportunity to remind you to get comfortable with looking at this type of chemical equation and the discussion that we are going to have about these types of chemical reactions. In most cases, you will not be required to memorize a structure or memorize the name of a reaction or a component, so a substrate or a product, unless it is a critical component in a pathway. When that occurs, I will make it very clear to you, you need to know this substrate, you need to know this product, okay? So make sure that is very clear in your mind. Now, the main reactions are redox, and this is a subscript for reduction and oxidation. So remember, reduction is gaining and oxidation is loss either of an electron or a proton. These are very common reactions. You have group transfer where you actually have a group, might be a hydroxyl group, it typically might be a phosphate. You take phosphate and you transfer it from one molecule to another. Hydrolysis, this is just where water, you see that in the diagram below, is used to break a bond. You've got we'll talk about those in a moment. Isomerization, which is where you've got different isomeric forms of a product or substrate being generated. And then you link groups together, and this is what ligation is. And so you'll see these in the tables as we're coming through, but there's also this diagram here. So this is our peptide. Here is our peptide bond. We can tell that these are amino acids because we've got our central carbon, proton, carboxyl group, and amine group. And here is our R group. We've got the same structure. So this is our peptide bond. The hydrolysis, and remember from the peptide bond we released water. So to break this apart we need to add water, and you can see here carboxyl group is regenerated and the amine group is also regenerated. So this is an example of a hydrolysis reaction where you need to break things apart. So the water molecules disrupt the bond. So here is the table that I was referring to. Oxidoreductases, so this is the redox. So it's the other way around. Rather than redox, they've written it as oxidation reduction. Doesn't matter, it's the same thing. You can see that this is a redox reaction. So oxidation, reduction. We've got here lactate is the substrate and the enzyme is a dehydrogenase. What this means is that it removes a hydrogen atom. The other thing that I want to remind you is that when you see the suffix ase, you know that that is an enzyme. So whenever you see the word ase, it is referring to an enzyme, in most cases. The next one was transferases, and remember, what does it do? It transfers a group. And so here you've got a kinase. We won't worry about the name at this stage, but a kinase basically just adds phosphate to a target. That's all it does. The other example are hydrolases. Remember, this leads to a hydrolysis reaction, so using water to break a bond. We saw that with chymotrypsin, so this is part of the digestive process. Lyases, here we've got the addition or removal of groups to introduce double bonds to make them more reactive or not. So this is fumarase. None of these are critical as an example to remember, just using them to tell you what's what. Isomerases give you that isomerization, so that rearrangement. That's what it's talking about here, group transfer. And you can see here, triose phosphate isomerase. It's going to convert the structure from one to another but keep the bonds together. And then the ligase, which links or ligates two substrates at the expense of ATP. So it needs this for energy. And here we've got amino acyl tRNA synthetase. This is part of protein synthesis. So make sure you get comfortable with all of these, because we're going to see these throughout the semester, throughout all of our lectures, so you need to be familiar with what is occurring. Okay, I'm going to stop there. When we come back, we'll start to talk about thermodynamics and how this interacts or interferes or is involved with enzyme activity. Okay, welcome back. So we finished talking about the different types of reactions, and now we're going to need to move on to talk about enzymes and thermodynamics. Now, you would have heard of thermodynamics in chemistry and the term of Gibbs free energy, and it's referred to as delta G. And so this delta G is a thermodynamic property, and what it does is it tells us whether a reaction is likely to proceed and be spontaneous or not. So can it occur on its own? That's the question. So when the delta G value is negative, so less than 0, so it's negative, a reaction occurs spontaneously. So when you have a negative value, it occurs spontaneously, and it's called exergonic. This reaction releases energy, and it's used to do work. So this is the term work here is to catalyze a reaction to generate a product from some biochemical perspective. On the other hand, when delta G is positive, it requires the input of energy. This can either be heat, but more typically, it's going to be through the hydrolysis of ATP, so the energy molecule. So this is called endogenic, and as it says, requires the input of energy. We'll get to the diagram in a moment. Now, the good thing about this type of sort of description is that once you understand the first one, the second is going to be the opposite. So exogenic, spontaneous, doesn't need energy. You know that the other one is going to be endogenic, doesn't occur spontaneously and requires the input of energy. And we will see both of these when we come to talk about metabolism and allowing processes to occur. So where does this free energy come from? It comes when you've got the potential energy that is stored in covalent bonds of a chemical product. And that energy is less than the potential energy of the starting substrate. So just think of it this way. The amount of energy in what you have made, the product, is less than what you started with, substrates of a chemical product. And that energy is less than the potential energy of the starting substrate. So just think of it this way. The amount of energy in what you have made, the product, is less than what you started with. And so if it is less, some of that energy is going to be released, and that process can go forward, and it makes sense. If it's the other way, you're going to a state where there is higher energy. For that to occur, you're going to have to put energy into the system. Now, if energy is released, this free energy can either be captured, can be stored, which is essentially the same thing, and it can be used to drive endogenic reactions on demand. Remember, endogenic require energy. So what this allows us to do is to couple or link together one reaction that might release energy, and that energy that is released by the first reaction can drive the next reaction, which requires the input of energy. So you can see it's a very nice balance by linking those two together. But irrespective, for any given reaction, an energy barrier has to be overcome for the reaction to proceed. And this energy barrier is called the activation energy. Now, the lower the activation energy, obviously the more spontaneous it's going to be and the easier for that to occur. So the energy required to initiate that conversion of reactants, the substrate is another term, so reactants is the other term, but substrate to products is what determines the rate of the reaction. And another simplified way would be the speed. But remember, rate refers to activity per time. So it might be that you've got 5 reactions per second. That is the rate. Okay, it's an important idea, an important concept to understand. Here's a nice diagram which shows you exogenic reactions releasing energy and endogenic reactions require the input of energy. So what it's showing is that if your starting point, so sorry but the diagram uses reactants, but substrate has a very high energy, to go down to the product, it's very easy. So it's like riding your bike down the hill. So it's negative because there's less here than it is up here. When it's the opposite, when your reactants or substrate generate a product which has higher energy, it's like riding a bike up a hill. You need to input energy. It's endogenic, and so this requires energy for this to occur. So it's a nice simple diagram, but it's really quite a really good example of the difference between the two. So when we come to talk about enzymes, we're going to be in both of these situations. And the last comment is you can imagine if these are all linked together, substrate goes down, there's energy, it comes to a product, that product now turns into the next reactant, and we want to go up, the energy that is inherent here allows the bicycle to keep going, go up, and generate a product. So you can see how the energy that is generated from one reaction that is exergonic can help to drive an endergonic reaction here. If this is not 100% clear to you, write a comment down, make it clear. When we come to the tutorial, we can go through this again. So what do enzymes do? Enzymes lower the activation energy of a reaction. So remember, this is the energy to start it. How do they do that? They stabilize what is called the transition state complex. And this is the intermediate. So what we normally see is that we have our substrate, and this is just shown as S, plus our enzyme. First of all, come together to give you this intermediate, which is substrate and enzyme. And then ultimately what you have is product plus enzyme. This is the transition state, where the two are very closely bound. The reaction is about to proceed, and the substrate then gets converted into the product. So enzymes only affect the rate of a reaction. This speed, by changing this, you can ultimately affect the rate of the reaction. And importantly, when we reach equilibrium, there is not going to be a change in the concentration of the reactants because delta G is 0. So this is important. You want to drive a reaction, you then need to either change the concentration of the reactant or the product. If these are equal, it's like the bike is on a flat plane. There's no difference. So by changing the concentration, we can change the location. So here is an example. Diagram shows the effect of an enzyme on the reaction rate and the time to reach equilibrium compared to where there is no enzyme. So this is product. So we're starting with substrate and we're looking at how much product is being generated. So you can see that with an enzyme, it goes up very, very quickly, and then it starts to reach equilibrium and plateaus. Without an enzyme, it's much lower, the amount of product is lower, and it takes a lot longer before it actually reaches this, and it may never reach that threshold. It's just going to go along very, very slowly, so you can see the massive difference. And this time is something that we spoke about earlier in terms of accelerating this process and allowing it to occur. This is another diagram to talk about the transition state and also the activation energy of a reaction. So here we've got the free energy and the progress of the reaction. So what we can see is we start off with our substrate, and here is our product. So we know that this is going to occur because the substrate has more free energy than the product. So this is going to be exergonic. We still have to get over this transition state, so this activation energy. If it is uncatalyzed, we follow the solid black line, you can see that the gap, the height of this peak is much higher. So the amount of energy that needs to come in is going to be greater. Enzymes, so the catalyzed state, reduce this. And so the energy, which might just be the heat in the system, so our own body temperature, is enough for this to occur. And so as it gets over this point, it's going to come down. And this is the delta G for that reaction. It allows it to occur. And you can see that you've gone from a substrate to a product which has less energy. So it's that negative value. Okay, let me stop there because I want to move on to talk about enzymes and their structure in the next part of this recording. Okay, welcome back. So now that we know what enzymes are doing, let's look at the structure of an enzyme. So we know that enzymes are proteins, and so they're going to be functional when they're folded into their 3D tertiary structure. And this is where we're talking about obviously a single chain. Many enzymes will also have a quaternary structure, but for keeping things simple, let's just imagine it's a single protein that's folded in its three-dimensional tertiary state. So the catalysis, this is the term which is the process of converting a substrate into a product. That's why it's called a catalytic event. Where does this take place? It actually takes place within a very special three-dimensional pocket or cleft called the active site of the enzyme. So you need to know this. You need to know the characteristics of the active site. First part is that it's actually a very small part of the total volume of the enzyme. It's not very big in comparison to all of the remainder of the protein. But when we look at this location, it has key amino acids that are important for the formation of that transition state. And these amino acids are the one where the catalytic reaction takes place, which might be a redox, it might be an isomerization, it might be a ligation reaction, one of the 6 that we spoke about. What we typically see is that because it's a small, very tightly packed area, water is excluded from the active site unless it's actually part of the reaction. So if this is a hydrolysis reaction, you need the water to be in there. Otherwise, the hydrolysis reaction won't occur. But if it's any of the others, the water can actually impede the reaction, so you need to, or the cell needs to exclude it from that site. When we look at the substrates, as they're coming into the all very tightly packed area, water is excluded from the active site unless it's actually part of the reaction. So if this is a hydrolysis reaction, you need the water to be in there. Otherwise, the hydrolysis reaction won't occur. But if it's any of the others, the water can actually impede the reaction, so you need to, or the cell needs to exclude it from that site. When we look at the substrates, as they're coming into the active site or the active pocket, how are they bound? They're bound by multiple weak interactions, and we saw this in the very first lecture. We've got ionic bonds, so charge, hydrogen bonds, and the non-polar van der Waals forces. Remember, these are all weak, but there are multiple interactions, so that helps to keep the substrate in its correct shape and configuration for the enzyme to work. So that's well and good, but you might be thinking, what about all of the other amino acids? What do they do? They serve mainly as scaffolding. They allow the protein to have a particular shape, the active site to have a particular shape to facilitate the reaction. But these other regions can also be involved in regulating catalytic activity. So you can think of it a bit like an on-off switch. They may allow the reaction to be turned on, or depending on those other regions, you might be able to turn the assay or the enzyme off. And we'll talk about this later. Now, there are 2 models that explain or exist and try to explain how a substrate actually binds to an enzyme active site. And you need to know both of these. There's the lock and key model and the induced fit model. Now, the lock and key is just as the name suggests, there's an exact match between the substrate and the active site. So, like a key will fit perfectly into its requisite lock, the substrate fits perfectly into the enzyme, and there's a perfect match. In contrast, the induced fit says that the enzyme has a much broader or general shape, and that after it folds, it now fits the shape of the substrate. So it has to conform and sort of adapt to the shape of the substrate. There's still some debate, but it's more than likely that the induced fit is probably the more correct of these 2. Now, if you look at this diagram, we talked about it earlier, but I'll just get to it now. This shows an enzyme in its tertiary state. So what you can see is that there is one protein. So therefore it's the tertiary state that is shown. It is fully folded, and you can see these individual amino acids. So we've got yellow, purple, green, orange, and red, and blue. Okay? Why do we highlight those? These are the active amino acids in the active site. So these are the catalytic amino acids. And so you can see that these are all quite close to each other. But if we took this protein and we linearized it, we made it straight like a piece of spaghetti, notice what we see. They're actually distributed throughout the protein. So what is the importance of that? What this is telling you is that these active site amino acids can be distributed, but when they fold together, they are brought closer together and they're brought into a particular configuration that allows the substrate, for example, to come in. And the remainder of these amino acids are just to ensure that these catalytic amino acids are in the correct configuration, are in the correct shape. And that's really important for the substrate to come in. So here is the first example. This is the lock and key model. Here is our substrate that has a particular shape. Here is our enzyme, and here is the catalytic site. Now, what I want to just highlight to you, A, B, and C, that these are amino acids, and these are your catalytic amino acids, similar to what we just saw in the previous diagram. And when these two come together, you can see that there's a perfect fit. This is the enzyme substrate complex, and so this is also your transition complex before the product has been generated. So you can see that there's a perfect fit. In contrast, the induced fit, you can still see the three catalytic amino acids and the substrate. The substrate hasn't changed, but the enzyme looks a bit less refined. It's a bit more fluffy, if I use the term. Not the most scientific, but you can see that it's not as defined. When these two come together, there is a shape change in the enzyme to give you this particular configuration. And again, this allows for the product then to be acted upon. And you can also see that the localization of the three catalytic amino acids, here they're much more far apart. And when you have the folding with the substrate, they're now in the correct configuration. And as it says down the bottom, B is most likely the correct model of the two. So let's think about substrate and orientation. Here's another diagram. So here is substrate, here is the enzyme, and this is the active site. Because remember, that's the bit that we're really interested in this part of the lecture. So here is the substrate. It has a shape, it has hydrogen bonds, it has charge, it has different molecules. This substrate can only go in this particular orientation because the cleft matches, so the shape of the pocket matches, but also we've got hydrogen bonds occurring. We've got ionic interactions occurring, and again, we've got hydrogen bond occurring here. If we were to rotate this 180 degrees, so this was on that side, you could see that it wouldn't fit. It wouldn't fit because this is squared off and this is rounded, but also we wouldn't be able to make these interactions. It wouldn't be able to make this hydrogen bond between the NH group and the oxygen. That wouldn't work. So the substrate can only go in one orientation, and as it says here, side reactions are therefore eliminated because the substrate can only go in one way. You can only get one product being generated, much more efficient. Okay, I'll stop there because the next part of the reaction goes into this a bit more. Next part of the lecture goes into how we can regulate activity. So it's taking what we've already learned, but now applying the regulatory or the control mechanisms that are in place. Okay, welcome back. So now we're going to move on to talk about regulating enzyme activity. So we spoke about it earlier when we said the active site, and I said to you that all of the other amino acids can have some regulatory control. So we're going to talk about how enzyme activity can be regulated. And there are four main ways of regulating enzyme activity. So we'll go through all of these: allosteric, multiple forms, reversible modification, and proteolytic activation. So allosteric control is quite an interesting one. Allosteric proteins have different regions, so distinct regulatory and functional sites. So regulatory, so this is the control site, and the functional site, this is really the active site of the enzyme. So when you have binding of a regulatory molecule to the regulatory site, it causes a shape change, and that shape change is called a conformational change. Now the shape change of the enzyme alters the active site. And so remember, you've got the 3D shape. The 3D shape keeps the active site in a particular shape. If you alter the 3D shape, you're going to alter the active site. So therefore the substrate may or may not be able to enter by altering the three-dimensional shape. So it's an easy way to control enzyme binding. And so what this is referred to is cooperativity, which means that activity at one site can affect the activity at the other site. And as a result, small changes in substrate concentration can be produced, or can produce substantial changes in activity. So whatever affects the shape, you don't need a lot of that regulatory molecule to therefore change the active site, and by doing so, the effect on the substrate is going to be massive. So only a little bit of binding at the regulatory site dramatically impacts the active site. So that allows for an amplification and also very fine control as to what is going on. The next idea is feedback inhibition, and this relates to allosteric control. As an enzyme acts, what you find is that the substrate is obviously going to decrease and the amount of product is going to increase. Now, unless there is some way for the enzyme to be controlled, you could have excessive amounts of product being generated. So this product can actually reduce the activity of the enzyme, and this is referred to as negative feedback inhibition. It's negative because we are turning off the enzyme. We're turning off the enzyme activity. So it's a nice control mechanism. Now here's an example, aspartate transcarbamylase, ATCase. This catalyzes or converts or leads to the condensation of aspartate. So condensation is really just like a ligation where the two come together. So aspartate, amino acid, and carbamyl phosphate. This is CO2 with a phosphate. It links those two together. This ultimately leads to the nucleotide CTP being produced. So this is one of the bases in DNA and RNA. This product, CTP, can inhibit the activity of the enzyme. So the end result is that it blocks its production. Now, CTP is actually structurally different from the substrate right at the beginning. So the question might be, well, how does it do it? If it doesn't bind, it's not binding to the substrate, how is it inhibiting? It's acting as an allosteric inhibitor. It binds to a regulatory site elsewhere on the enzyme that affects the enzyme's active site and effectively turns it off. Okay, so it's a beautiful example of allosteric control. The next example, or the next process, is having multiple forms of an enzyme. And so what you can have are modified forms of the same protein being produced. And these are called isozymes or isoenzymes. Same thing, just different names. So these catalyze the exact same reaction, but will differ slightly in their structure as well as their regulatory properties. And what you find is that they're often expressed in distinct tissues, organelles, or even during different stages of development. So you might have one protein being made in utero, then as a baby you switch to a different enzyme, then as an adolescent another, and then maybe another as an adult. That's another way of doing that. And you could change this. You could say that this could be a different tissue. So it might be the spleen, you have one version, you might have another version in the kidney, the lungs, and the heart, as an example. Okay, so same idea, same enzyme, but slightly different and acting in a different location. One that we will see very often is this one here, reversible covalent modification. And this is where the activity of an enzyme is modified by covalently attaching a group to the enzyme. And the most common way that this is achieved is to add a phosphate group from ATP. So it says here phosphoryl, but essentially it is that, a phosphate group from ATP. Addition and removal of phosphates are probably the most common way of modifying a protein or an enzyme. So the transfer of the phosphate group generally, not always, but usually turns on or activates the protein, and therefore removing it will deactivate it. Now there are situations where we've got the reverse, but this is typically what we will see. This reaction is catalyzed by an enzyme called a kinase, and you can tell it's an enzyme because of the ASE, and it's removed by an enzyme called a phosphatase. If you want to know more about this, go to the signal transduction lecture when they're released, but that's not till much later in the semester. But what you'll see here is that the kinase adds the phosphate, and the phosphatase removes the phosphate. So a very simple way of regulating activity. And the last mechanism is proteolytic activation. And the idea here is that enzymes require activation after they've been synthesized and have folded correctly. So what you also will see is that some enzymes are made as an inactive precursor, and they have to be cut or cleaved by one or a few peptide bonds for full activity. So they're made, pre-made, ready to be stored but not having activity. You then chop off a terminal portion, and by removing that terminal portion, you convert it from an inactive to an active state. And the term here is a zymogen or a proenzyme. And again, we will use these terms interchangeably. So, zymogen, you can see that we've got pepsinogen. You can tell that this is the precursor. It's cleaved into pepsin. Chymotrypsinogen is chymotrypsin, and trypsinogen is converted to trypsin. So these are those precursors. I already hinted as to one of the benefits is that these can be stored and then activated quickly. You don't have to go through the whole process of transcribing the DNA, translating it into a protein, modifying it, making it. It's already there, so you can then convert it very, very quickly into the product that you require. Okay, I'm going to stop there. This was a reasonably long and complex lecture, but some very fundamental concepts of enzyme activity. So go through the chapter summary, make sure you put down your own questions, make sure you go through the tutorial questions to cover this content, because this is really at the heart of biochemistry, and we're going to go through a lot of these concepts repeatedly, but in a different perspective through metabolism. Okay, see you at the last lecture.